Welcome dear students, hope you're all doing fine. Today we are going to talk about sterilization and disinfection by chemical methods. That's the part 3 of the series. In part 2 and 1 we have discussed about the physical methods. So let's talk about this now. So this class is intended for MLT second year, ACOT second year and ACOT third year. So let's begin. As far as sterilization is concerned, we have been talking about the physical methods in the previous 2 lectures. So it covers all this portion over here. So we have talked in physical method of sterilization is sunlight, heat, vibrations, radiations, and filtration. So now today we'll be particularly focusing on the chemical methods. So it includes this part. Okay. So in this we'll be talking about the chemicals which are in the liquid form, for example alcohols, aldehydes, phenolics, halogens, heavy metals, surface active agents, for example detergents, dyes, gases, for example formaldehyde, that is HCHO, then we have ethylene dioxide, and we have also the plasma. In physiochemical methods, it is a combination of the physical and chemical methods where we'll be using steam as a form of a physical method and formaldehyde as a form of chemical method. So that is what we know by physiochemical methods. So let's move on. So when we are talking about chemical methods, you should remember that chemicals or many chemicals are used as disinfectants, and we have to classify them one by one, and we'll study them inshallah in particularly much detail. So let us come to the point and talk about this disinfection. So in case of disinfection, we will talk about disinfectants. So what are disinfectants? Disinfectants are those chemicals which can destroy the pathogens. And destruction means either you can say you can destroy them, stop them multiplying, or you can sterilize them completely by killing them. So disinfectants are particularly used on inanimate surfaces, for example tables. They can be used on doorknobs, which we usually touch often. So we can also use these chemicals. Now they can have different range of activity. They can have a narrow spectrum activity, or they can have a broad spectrum activity. This means narrow means it will be specific for only one microorganism. For example, it can be only specific for fungi. So we call it as narrow range of spectrum. In case of bacteria, for example, if a drug or your chemical works only on mycobacteria, so we will also call it as narrow spectrum. So it doesn't act on other bacteria. Okay. So in case there are also some chemicals which have broad range of spectrum, and one of them I can say here is there are for example different kinds of disinfectants that can be used, which have a broad spectrum activity. It can be active against viruses, it can be active against gram-negative bacteria, it can be active against gram-positive bacteria, it can be active against the other fungi and parasites. Okay. Okay. Now the chemicals that particularly sterilize can sterilize your microorganisms or the place where you are working on, we call them as chemisterilants. Okay. So sterilization means completely destroying the organisms which will not be there. It means killing. So another term which we have been previously using for SIDL drugs, for killing, is SIDL. Okay. And static means it prevents their growth. So those chemicals which can sterilize, it means they can kill or kill the bacteria, we call them as chemisterilants. Those chemicals can be can be safely applied over the skin or the mucous membranes. For example, our inner mucous membrane of our mouth, we call them as antiseptics. Okay, the same disinfectants which we can use on the skin or mucous membranes, we call them as antiseptics. Okay, So what are the different features of an ideal antiseptic or a disinfectant? So these are the following properties. Let us see one by one. They should have a wide or broad spectrum of activity. It means that they should be active against different kinds of microbes. Okay. Then they also should destroy the microbes in a particular period of time. So it means destruction should be done, or it should be effective, and should kill bacteria very fast. That is what is the practical period of time. Now they should be also active in the presence of the organic matters. And organic matters are whatever you eat, for example your rice and any organic matter, potato, when you eat them and throw them out, and in the presence of these organic matters, your disinfectant or antiseptic should be active. So another feature that should make an effective disinfectant is they should have an effective contact with the microorganisms, and they should be wettable. Wettable means they should wet up the microorganisms so that it comes in close contact with them. Okay, now talking about continuing these disinfectant properties. So they also should be active at different pH levels, either acidic or basic. They should be stable for a longer period of time. They should have a long shelf life when you are not using them, when you are putting them in your cupboards, and they should be speedy in action. It means they should act very fast and kill the microbes or stop them multiplying. So they also should have high penetrating power, because many bacteria, for example gram-positive bacteria, they have a very thick cell wall called peptidoglycan. So they should penetrate this wall also. This is just an example. They should be non-toxic to our cells. It should not cause us any allergy, and it should not be irritative for us to use, and it should be non-corrosive in nature. What it should not be is, again, it should not have a bad odor, and it should not leave any volatile residues around or any stains. Another thing is they should be not expensive, so that any hospital can use them at an economical rate, and they should be available readily in the market. Efficacy should not be lost by reasonable dilution. Particularly whenever you use a disinfectant, you can dilute it with water. So if you dilute it with water, it should not lose its efficacy. But as far as ideal is concerned, it means the ideal disinfectant, it's not yet been available in the market. So what we are trying to maintain the balance is that we will maintain the balance between the concentration of the disinfectant. Okay, we can use the higher concentration, or we can increase the time duration of exposure of the disinfectant on the microbes. So we have to balance between these based on which microbe we are facing, or which microbial contamination is coming in your laboratory. So the level of disinfection can be achieved based on the contact time, how much time you need to put your disinfectant in that particular what you want to remove those microbes, at what temperature you are using them, then what type the chemical is, and what is this concentration. Can it work in the presence of organic matter, like whatever food we eat, and can it and what type of quantum of microbial load it can decrease. So by microbial load, particularly we use the term called as logarithmic growth. Logarithmic means, for example, if I say 10⁻1 is 10, okay, and 10⁻2 is 100. So microorganisms have the ability to do logarithmic growth. Okay, so they don't multiply by 2 and then 4 and then 8. So they multiply by a logarithmic growth. So it should be how much load you want to shed of the bacteria or any microorganisms out of your working place. Now the chemical disinfectants, it should work at concentrations. When you use it at a particular concentration, it should not lose its strength, or it should not lose its length when you keep it standing for a long time once you have prepared the disinfectant. Okay. Now classification of disinfectant is based on whether they are liquid, okay, whether they are gases. So it's based on whether they are liquids, whether they are liquids, whether they are gases in nature. They are also classified based on their spectrum of activity. Do they have high level of activity, broad spectrum on many microbes, or intermediate or low activity? They can also be classified based on their mechanism of action. For example, some of the chemicals, disinfectants, can act on the membranes, particularly in gram-negative bacteria or viruses. They have a membrane surrounding it. So in case of this, alcohols and detergents can be used. Now another mechanism of action that should kill the microorganism is denaturation of the proteins. So denaturation simply means that if you have a particular enzyme that has a three-dimensional structure, and when you heat it or when you treat it with a chemical, it loses its three-dimensional structure, so it's becoming non-functional. So that's what you mean by denaturation of proteins. Okay. So another way of killing the microorganisms using chemicals is oxidation of essential sulfahydryl groups present on the enzymes. You will see that when SH is present on enzymes in a three-dimensional structure, for example, here and here we have two sulfahydryl groups. Okay, so these are sulfahydryl groups, just like your hydroxyl groups. Only the difference is they have sulfur here in replacement of oxygen. So let us say that this is a covalent bond, sulfahydryl bond. So what it does is it can break this sulfahydryl bond and denature your enzymes or proteins. So this is how they work, and this is particularly done by, for example, by hydrogen peroxide and halogens. For example, chlorine and your fluorine and your silver, etc. Another way of mechanism of killing the microorganisms is alkylation. Alkylation is adding the alkyl group, that is CH3 C double bond O. So it gives a negative charge on the surface of either the amino groups, that is NH2, or your carboxyl groups, that is C double bond OOH. Okay, or your hydroxyl groups. So when it's alkylated, the bonds are broken down because of the negative charge, and this is done by the gaseous compounds, for example, ethylene dioxide and the formaldehyde, that is HCHO. Very simple in structure. They can also kill the microorganisms by damaging their nucleic acids. For example, they can damage the RNA, or they can damage the DNA. Many RNA containing viruses, DNA containing viruses, okay, they can damage them and kill the microbes in that case. Again, these two gaseous compounds can do this DNA damage. Okay, moving ahead. Now we have alcohols. Alcohols simply means you have any compound. Okay, so it means you have any compound. So for example, this, and you have a hydroxyl group attached to it. We call them as ethanols. Now based on this R, we can have this CH2OH, that's called ethanol. Ethanol is one of the most used alcohols in your research laboratories or your diagnostic laboratories. Now mode of action of these alcohols is they dehydrate the cells. It means cells have, most of the part of the cell is water, so it dehydrates them, that water gets out from the cells and they can die and they can shrink out. They can also disrupt the membranes, that is, for example, if we have a virus like this, okay, they have an outer membrane or coating around it, so they can destroy this membrane and then kill the viruses. They can also do the coagulation of proteins. So you might have heard about blood coagulation. It's done by your platelets. Okay, the same happens with case of the proteins. So the protein is functional one here, and they come together and they coagulate, so they no longer work, they accumulate, and that is called coagulation. So three ways now: dehydrating cells, disrupting the membranes, and coagulation of proteins. That's the work alcohols do on the microbes. So best example is ethyl alcohol, which I told you also, and in most of the cases we have also isopropyl alcohol or methyl alcohol. So methyl alcohol is simply CH3OH, isopropyl alcohol has 3 carbons, and ethyl alcohol, as I told you, it's like CH2OH. Okay, it is okay. So now applications are we use 70% of the alcohols, particularly while you are working in your diagnostic laboratory, or where if you are doing some work in your biosafety cabinet. So you clean it with 70% alcohol, that is 70% ethanol. So why 70% is particularly being used? There's a reason for it. So if you use a concentrated form of alcohol, what you can do is, for example, this is your microbe, okay? So when you use 100% alcohol, or 90% or 95%, it does coagulation of proteins on the surface, and this doesn't allow this your chemical sterilisant or alcohol to penetrate inside and coagulate the bacterial or viral enzymes. So one reason is that's why we use 70%. It means you are diluting it with for every, if you are preparing 100 ml of your ethanol, so what you will do is you will take 70 ml of ethanol, that is sprit, and you will add around 30 ml of your distilled water to it. So it prepares your 70% of ethanol. Okay. So another reason why water is being added to the ethanol to make it 70% is because ethanol vapors, they evaporate very fast. So when you add water to your ethanol, so it becomes less volatile. Okay, it has less evaporation, so it stays in close contact with the microorganism. Okay. So another name of your 70% ethanol is sprit, and it can be used as an antiseptic on the skin. Once you do finish your work on the microorganisms, you can just come out of your hood and put some your alcohol on your hands. It just acts as a disinfectant, where particularly we are using, particularly in this time, in coronavirus time, we are using so much of antiseptics, or you can say your disinfectants. So now another alcohol that can be used is the isopropyl alcohol, and it is preferred over ethanol in cases where you have to disinfect surfaces, or you have to disinfect the thermometers which you are using time and again for checking the temperatures of the patients. Now another kind of alcohol, that's methyl alcohol, that is CH3OH. It particularly kills the fungal spores. Okay. So for example, if you have a fungus growing on a skin and it forms spores like structures, so it can kill those, that's methyl alcohol. Okay, so hence it's very useful for disinfecting the inoculation hoods, where inoculation means where you are using microorganisms. So when they are being used, you should usually you dip them into this methyl alcohol, or when you are using the inoculating loops, you can dip them in the methanol before using them again for inoculation. Okay, so what are the disadvantages of these alcohols? They can be irritating to your skin, they evaporate very fast, and they can be inflammable. So this is one of the most important disadvantage of your alcohols, and you need to be very careful. So while I was working in the biosafety cabinet once, and once I rubbed cleaned my surface with the 70% ethanol, my sprit lamp was here, and it immediately caught fire when it came in contact with the cotton, and it can cause fires, outbreaks. So you should be very careful while using alcohols as your chemist learns. Okay, next. Now next, what we are talking about is what we can use is, these are the aldehydes. So aldehydes, usually one of the aldehyde, we have talked about different kinds of aldehydes. They have this formula, that is R, C, double bond O and H. Okay, R can be any group you can add here. So now again, the mode of action is alkylation. So alkylation is putting this group onto their amino endings. Okay, like C double bond O negative. So again, it adds a negative charge here, and it can act this and do this on the amino acids. It can do this on your carboxyl or hydroxyl. Carboxyl is OH, COOH. it can do it on your hydroxyl groups of any microbial enzyme. So once alkylation takes place, then usually what happens is your enzymes don't work properly, and this leads to their killing. Again, another mechanism of killing of microorganisms by aldehydes is by damaging the nucleic acids. So that either by either RNA nucleic acid or nucleic acid or D-nucleic acid, that's deoxyribose nucleic acid. Okay, and thereby kills. It means it's a still in function, and it also includes spores. Okay, so there are many gram-positive bacteria which form spores. One example I can give is Bacillus anthracis. Bacillus anthracis is a spore-forming gram-positive bacteria, and it causes anthrax. Okay, so once this anthrax spores were being enveloped and used in the form of bioterrorism, and they were delivered to the president of USA of that time. So these anthrax spores are an example of the gram-positive bacteria. And examples that what kinds of aldehydes can do that, it's your formaldehyde and your glutaraldehyde. So what are the applications of your aldehydes? Number one of the application which we particularly use in your laboratory when you are working with your microorganisms is fumigation. So fumigation means you add some chemicals and they fumigate your lame. It means fumes come out and it disinfects either your rooms or your chambers or even your operation theaters. Okay, so this is being done, but there should be no human being inside, and you can do it in different places. So how do you prepare this? 40% formaldehyde in the form of formalin is being prepared, and then it's used for disinfection and particularly fumigation as I told you before, in operation theater rooms and biosafety cabinets and patient wards in the absence of the human beings. Okay, so this fumigation can be achieved by different ways. Either you can prepare this 40% formalin and let it boil on. So this is one way. Another way is you can heat the paraformaldehyde. This is the second way. And the third is if you treat this formalin, once you prepare this 40% of formaldehyde, that's called formalin, you can add potassium permanganate, and that is K2MnO3. K2MnO3, potassium permanganate. So it's like it's very brownish or reddish, you can say. It's like pink in color. Okay, so once you add it to your formalin, it will start fumigating, and it will start forming like a smoke in the place where you use it. So what you can do is you can use a Petri plate. Okay, put a formalin inside and then add to it the KMnO4, that is potassium permanganate. Okay, so where it's used for sterilizing beddings, furnitures, and in libraries for books. Another way of another example of these aldehydes, two examples are formaldehyde and your glutaraldehyde. So we can use formaldehyde in different concentrations. Previously we used 40% formalin, that's called 40% formaldehyde, so it formed formalin. Okay, so now you can use it in different concentrations. See, 10%, 2%, 2%, and you can use them in combination with other compounds. For example, if you add 10% formalin to 0.5% of tetraborate, it can sterilize and clean metal instruments. If you use it, you use glutaraldehyde in a concentration of 2%, means you will use 98% of water, distilled water, and you will add 2 ml of glutaraldehyde to it. So you can get 100 ml of your volume of 100 ml of 2% of glyceraldehyde. So depending upon your need, you can increase the volume. So you have to change this and this. Okay, you have to increase both in a same ratio. So they can be used for sterilizing the thermometers or cytoscopes, your bronchoscopes that you use for lungs, centrifuges where you use it for clearing out the cell organelles or any other thing. You can also use it on anesthetic equipments which are used in the anesthesia and critical care. And now again, as I told you, when you use different percentages, it depends upon the exposure time. So you can use it for at least 3 hours, and at an alkaline pH, it means pH should be either 7 or it should be greater than 7. Okay, and this is required for the action by the glutaraldehyde. When you use 2% of formaldehyde, you keep it for 40 degrees Celsius and let it stand for around 20 minutes. So this is used to disinfect wool and other cotton, anything you want to use before your surgeries. Okay, so now see, if you decrease the concentration, now here you use 2% and now you are using 0.25%, that's almost 1/4 in this case, or in this case it's around 1/8 concentration. So but then you have to increase the temperature. See, here you are using 40, here you are using your 60, and you are also increasing the duration of exposure, that's 6 hours and compared to the 20 minutes. Okay, so when you use it in this concentration, you can disinfect animal hair, you can disinfect the bristles, which you use for cleaning different things. Now there are every chemical or every disinfectant has some disadvantages. So what are the disadvantages of aldehydes? That the vapors can be irritating to you. That's why most of the time they are being neutralized by addition of the ammonia, that's NH3. Okay, it has poor penetration power, so it cannot penetrate most of the viruses or the gram-positive bacteria which have a thick peptidoglycan cell wall. It leaves some volatile residues after using it, and its activity can be reduced in the presence of different kinds of proteins. So glutaraldehyde also requires alkaline pH. If the pH is means acidic, it is if it's around 2 or less than 2. So in these conditions, it cannot work. So only those articles can also be sterilized which are wettable. Okay, which cannot be wettable, so they cannot be sterilized. It means which are, for example, which are hydrophobic in nature. They cannot, absorb this. Okay, so the sterilization will not be achieved. Another way of using chemical method is phenol. So phenol is simply a ring-shaped structure. You might have studied it in class 11, 12 in your chemistry, in organic chemistry. So this is your phenol. Okay, so then you can convert into different forms based on the derivatization of this phenol. So what is happening is, how does they work? Again, they disrupt the outer membranes. For example, your coronavirus, okay, is an RNA containing virus. Fine. So then it has its own covering, that is every virus has a nucleic acid and covered by a protein coat. And then outside the protein coat, it has its outer membrane. That is how it gave its name corona, because it looks like a crown on the surface. It has these spikes around it, which RNA virus also has. So these spikes, help the coronavirus to attach to our nasal membrane cells. So we have receptors on our nose, inside the nostrils, and this helps the coronavirus to bind to our nostrils and then infect us. Okay, so it is very good for disrupting the membranes. Also, gram-negative bacteria, they also have gram-negative bacteria also have an outer membrane apart from peptidoglycan. Again, they can disrupt it and the gram-negative bacteria can be killed. Okay, second thing is it precipitates out the protein. So precipitation means in your solution they will be out from the solution. It means if you put protein inside, rest of the things will remain there, but proteins will precipitate and they will be out of, they will easily, you can easily observe that they will be out of the water, so they will not work again. So, and it also inactivates the enzymes which are used by the microbes for their day-to-day life. Now again, you can see the different concentrations and different chemicals can be used for different types of disinfection. So 5% phenol can be used, 1 to 5% cresol, 5% lysol can be used. It's actually the same cresol, but it's saponified. It means you add soap to it. Now you have also hexachlorophene, chlorophene, and hexachloridine, particularly when you go to the market and you want to buy hand sanitizer, it will contain chlorhexidine. And again, when you wash your hands with Dettol, that is chloroxylenol. So remember this, that Dettol, the concentrate of the Dettol is your chloroxylenol. Now applications, the first application that was used, where the phenol was used was by a surgeon called as Joseph Lister, and he used it to prevent any infections during the surgical interventions. Now phenols actually are core tar derivatives, and they you can say that they act as disinfectants at a higher concentration. So concentration should be very high, around say 98% or 95% concentration. But if you lower the concentration, they also act as antiseptics. Okay, they are acidyl, see, bactericidal, microbicidal, fungicidal. It means they are all, it has a killing effect on these microbes. Okay, but again, it is inactive against spores, as I told you about Bacillus anthracis spores or Bacillus subtilis spores, which are mainly found by the gram-positive bacteria. Okay, and most viruses, it is not able to do anything about those. So in case of coronavirus, you cannot use this phenol. So the corrosive phenolics are used for disinfection of ward floors. Okay, so you can disinfect your ward floors by using the phenolics, and also the discarding jars which are present in the patients or laboratories, and also the bed pans which are surrounding on the table of your patient. Okay, and then they are not readily inactivated by organic matter. It means they have this advantage. In presence with organic matter, for example, when you put something in your dustbin, you are eating some food and you have put them in the dustbin, and now you want to clean it from the microbes. So, but it has an organic matter attached to it, just like your kitchen waste. So it can readily inactivate the microbes in presence of the organic material. Okay, moving to the next slide. Examples of phenols you can use is chlorhexidine. So in case of chlorhexidine, it's actually an isopropanol solution. Okay, that it can be used in a form of isopropyl solution for in the form of an antiseptic for skin disinfection. You can use it as a hand sanitizer, and aqueous solutions, for example, for irrigation of the wounds. Again, it can be used as an antiseptic, as I told you, in the form of hand wash. Okay. Now again, we will see different concentrations and different percentages, as I've been talking from the very beginning in the physical methods also. So if you mix this chlorhexidine and make chlorhexidine gluconate solution, so it's used in pre-operative hands for cleaning the hands before the surgery is being done, and preparing the skin for the surgery. You can just wipe the cotton with this 20% chlorhexidine gluconate, and it's also used for general skin disinfection. So you should always remember that your skin is teemed with many kinds of microorganisms, but they are beneficial to us. Okay, but if there is another bacteria coming in our skin which is not or normal microflora, then it can cause diseases of skin. Okay, for example, fungus causes many kinds of skin diseases, so you can use it against these fungus. Chlorhexidine gluconate is actually mixed with quaternary ammonium compounds. So ammonium compounds NH4, which particularly form quaternary compounds in your chemistry. So example is citramide. If you add it to the citramide, and you get a stronger and broader antimicrobial effects. And usually it's present in the market in the form of Savlon. Again, you should know that Savlon contains antimicrobial effects. It can come in your exams. That's why I'm telling you. So when I'm stressing on something, you should understand. Okay. Now, what are the disadvantages of using chlorhexidine? So chlorhexidine is it's toxic, and it's corrosive. It can irritate the skin of many patients, and it's also inactivated by the soap. which is anionic in nature. Okay, so anionic means soaps which are negative charge. Okay, now talking about this chloroxylenolones, which are another derivatives of phenols, that is chloroxylenols. So chloroxylenols are less irritant, and they can be used for topical skin creams, and they are more effective against particularly gram-positive bacteria. So few examples of gram-positive bacteria I can give you is your Bacillus anthracis, which causes anthrax. Another example of gram-positive bacteria is your Staph aureus, Staphylococcus aureus, which causes many infections in the wounds. Staph aureus, A-U-R-U-S, Staph aureus. And you should know that they're both spore-forming bacteria, and they can lead to MRSA, that is methicillin-resistant Staph aureus. Methicillin is an antibiotic, and this Staph aureus once it becomes resistant to it, so it becomes a drug-resistant bacteria, and we have to look for alternative for dealing with this. Okay, so particularly drug resistance is coming up in this Staph aureus, which is very dangerous because it's particularly affecting the wounds. And another example of drug resistance, which is very prominent, which is very common around, is the drug resistance of drugs against Mycobacterium tuberculosis, which causes particularly MDR-TB. So this MDR-TB, it is resistant to two main drugs which are used in the TB treatment. One is rifampicin, which causes damage to your RNA polymerase in your in this Mycobacterium, so it cannot do any transcription, and it's also resistant to the INH, which is a cell wall inhibitor of Mycobacterium tuberculosis. So you can see that particularly it acts on the gram-positive bacteria. So Mycobacteria, it falls in no category. It's neither a gram-positive bacteria, it's neither a gram-negative bacteria. Okay, so this bacteria is called as acid-fast bacteria because it doesn't it's very resistant to the stains. Okay, so gram-positive stains, they usually dye. They're called gram-positive because while staining they color purple. They have a purple color, and when you try to wash it by acid alcohol, they do retain the stain because of their thick peptidoglycan layer. Okay, but it is not present. It's not effective against the gram-negative bacteria. So remember this chloroxylenols, they are not effective against gram-negative bacteria. Now we can again derivatize this chlorohexanol by different ways in chemistry. So if we have hexachlorophene, it's a chlorinated diphenyl. So it's like you use the halogen chlorine here, and you use 2 rings of phenol. So it is much less irritant because in case of many patients, the irritation, it's not good. So it has marked effect over the gram-positive bacteria, as I told you, as I gave you 2 examples of the gram-positive bacteria, that is Staph aureus and your Staph aureus, that is Staphylococcus aureus, and Bacillus anthracis, which are spore forming. Most of the gram-positive bacteria, you will find that they form spores around themselves so that they can survive even outside the host. And as I told you, they have poor activity against gram-negative bacteria. They cannot do anything to the Mycobacteria, not effective against fungal infections, and not also effective against the viruses. So when you find that the contamination is because of the gram-positive bacteria, in that case, you can use this. Okay, this is the one choice of your disinfectant. Now we also have triclosan. It's an organic phenyl. So it's organic phenyl ether with good activity against, again, the gram-positive bacteria. That is the 2 examples I gave you, and it's effective to some extent to a gram-negative bacteria. There are different kinds of gram-negative bacteria. So we have a bacillus kind of bacteria, we call it a E. coli, which is present in your gut. Okay, E. coli. So we also have another gram-negative bacteria that includes Pseudomonas P. aeruginosa. So another example of gram-negative bacteria is which are comma-shaped. For example, Vibrio cholera, which causes cholera in humans. Vibrio cholera. Another example, which is like a comma-shaped bacteria, like it is like this, and it has some flagella around it. That is called. It is present in your gut. I mean your stomach, and it causes gastritis. Gastritis. The name of this is Helicobacterium pylori, which is responsible for gastritis and the gases you have in your stomach. Sometimes you feel, and it's also responsible for cancer if it is not being treated properly. So people who are having gastritis, this is because of the Helicobacterium pylori. Okay, so patients who have these kinds of problems, stomach problems, they should do this H. pylori test. Okay, it has been found that it is involved in many kinds of stomach cancers. Okay, now what are the disadvantages of this chloroxylenols? So the disadvantage is it's inactivated by the hard water. Hard water means it is not being it's not the water that comes to your tap water. So hard water is present particularly in the wells when you use it. So it's a hard water. It's not being cleaned of the impurities. So in that case, it will not be active. Okay. Now coming to the halogens, one of the typical example of halogens which is used in cleaning water in the wastewater treatment, it is chlorine. Okay. Now, what's the mode of action? So, particularly halogens are oxidizing agents. So, oxidation means, for example, if there is an enzyme in a bacteria and it has a subgroup here, let us say an amino acid, NH2. Okay, amino group. So, what it does is it oxidizes this. It means it is losing the electrons. The microbial surface is losing the electrons. That's called oxidation, and because of this oxidation, this whole enzyme can disrupt and it becomes non-functional. Okay, so the cause halogens, what they do is they cause oxidative, and they damage the enzymes by oxidation, and particularly for sulfur groups. You will find that in many cases when proteins are bound by, if they have, for example, particularly in methionine and cysteine, they are sulfur containing amino acids. Okay, and they are present in many kinds of different kinds of enzymes. So what they do is this is a sulfur hydride. Okay, just like your OH hydroxyl group, this is another one. So they form a covalent bond. So sulfur hydride is a covalent bond. Okay, so they break this and this disrupts the enzyme and finally killing the microorganism. So chlorine, because it reacts with water, that's why you should understand why chlorine is also used in swimming pools to get rid of many microorganisms. So when it comes in contact with water, it forms hypochlorous acid, and in this acid is microbicidal. Again, cidal means killing. Okay, so it kills many of the microorganisms in water, in drinking water and in your swimming pools. Now chlorine compounds include, for example, chlorine itself, bleach, the household bleach, then the hypochlorite. Hypochlorite has a very strong smell and it vaporizes, and you should be very careful when you use or open the hypochlorite in your lab. Okay, so it has to be used in very less concentration. So it comes from the market as 40% hypochlorite. Okay, and it has a very strong smell, so be cautious about it. Now coming to the iodines, from the beginning, if you have sometimes been injured while playing as a kid, you will see that doctors used to put on the surface of the cotton, they used to put some iodine on its surface. Okay, and then put it on our wounds if they're a wound on our leg or hand. So this is called tincture iodine, where you have iodine. If you use iodine for on your wounds, and we also have iodophores. Now applications is tincture of iodine. Usually it's at 2% iodine and 70% alcohol. So what you have to do is, when you prepare 70% alcohol, what you do is, first example is, you can add around 70 ml of ethanol, plus you will add then instead of putting around 30% ethanol, you can use 27% of ethanol, sorry, 27 ml of your water, that should be distilled water, and then add to it again 2 ml of iodine. So this will iodine. Please bear with me, my handwriting is not so good, but inshallah I will try never to give a wrong concept. Okay, so this is tincture of iodine. You can prepare it this is your 100 ml. So according to your need, you can prepare. Now it's an antiseptic. As I told you, it can be used on the surface of the skin, on the wounds. So iodine can be combined with some neutral carrier polymers, for example polyvinyl pyrrolidone, to prepare iodophores. Okay, so one of the examples of iodophores is povidone iodine. So if you it's presently particularly in the mouthwash containing so if somebody is having inflammation of gums, a doctor prescribes him a gargle or a wash, and that has povidone iodine in it. So it's just like dark brown in color, as iodine usually has a dark brown color. So it's also dark brown. It's dark brown in color. Okay, so we'll find it in the given to the patients who have inflamed gums. So iodophores permit slow release, okay, and more slow release, more effective against the microbes, and reduces irritation. So it is particularly used as antiseptic. Antiseptic, again, you remember that it is either good for skin or applied to the mucous membranes. So mucous membranes are present in our oral cavity. So that's why povidone iodine is being prescribed for decreasing the inflammation of your gums. Okay. Now, if we talk about iodophores, they can again be used, as I told in the other lectures, they can be used in combination, or they can be used in different percentage. So iodophores can be diluted with 50% alcohol, and they can be then used for washing your hands. 10% again, if you use this povidone iodine, and when you use it, it can be used in the pre and post-operative skin disinfection. So whenever a surgeon decides to do a surgery, so he can use or he uses particularly this povidone iodine, which is undiluted. It means there's no water added to it. Okay. So chlorine gas, and this is used to bleach water. And in higher concentrations, chlorine can disinfect swimming pools, as I told you earlier. So household bleach can be used to disinfect the floors, particularly when we start cleaning the tiled floors. We use this disinfectant, and we use it particularly in the dilution of 1:10. So 1 part of your bleach and 10 parts of your water. Again, if you decrease the concentration, see, I again told you, if you decrease the concentration, this is only 0.5% sodium hypochlorite, okay? And it is used in serology and in virology. Serology means study of serum, which is present in our blood, part of our blood, and study of the viruses. Currently, you can talk about its study in coronavirus, okay, COVID-19. It was discovered in the 31st of December 2019. That's why the 19 comes here. Okay, so if used at a dilution of 1:2, 1:10, it can decontaminate and spillage of infectious materials. Okay, again, mercury chloride, if you combine chlorine with mercury, it is used as a disinfectant. Okay, and there are some disadvantages also while you use halogens. So these are rapidly inactivated in the presence of organic matter. So in case of cleaning your dustbins or other things, you cannot use their halogens, I mean this halogen containing compounds. Iodine also it's corrosive in nature, and it can stain. It has a staining problem. It can stain the place where you use it. And bleach solution is corrosive, where you use this halogen, and it can corrode the stainless steel surfaces, particularly when in your surgery. A neurosurgeon keeps all his equipments on a stainless steel container, so please don't use that in that, otherwise it can corrode the steel, and the infection chances are higher. Now, talking about their mode of action, they have done. So what they do is, as compared to the other chemicals, most of them either coagulate the proteins, or they add alkyl groups to it. So same in this case, it precipitates the proteins. So precipitation of proteins takes place. Proteins come together and precipitate, or for example, if they are in the solution, they can precipitate down and become non-functional. Now it can also do oxidation of sulfur-hydryl groups. So if there is a sulfur-hydryl covalent bond within an enzyme, it can break it. Okay. So by the process of oxidation, by removing oxidation, by adding oxidation, this bond is broken. So they are bacteriostatic in nature. It means they are not killing the bacteria, but they are keeping the bacterial load less. So example is mercury chloride, silver nitrate. Particularly use this silver nitrate on wounds, or in burns. So whenever there is a burn, you might have used a tube which has silver nitrate in it. Okay. So we have also copper sulfate. We have organic mercury salts, for example, mercurochrome and merthiolate. So applications of 1% silver nitrate solution can be applied on eyes. And also it was used for the treatment of ophthalmia neonatorum, but now it's not used. So this was the Creed's method, but it's not now followed currently. We also have silver sulfadiazine is used topically. It means it's used on your skin or mucous membranes to prevent colonization and infection of burn tissues. So whenever you have burns on your skin, so it means your skin is one of your defenses. It's a part of your innate defense system, and the skin is around this largest organ of your body as well. It measures around 2 square meters in length. So whenever his skin is bleached, there are chances, because there is normal microflora, and then it can, if there is no normal microflora around, so there are chances that pathogenic microorganisms can come and infect your wound. Okay, so we should be careful about this. Okay, now mercurialis. So now adding these different names to it, they are the same things but with different derivations. For example, mercurials are active against viruses at a dilution of 1:500 and 1:1000. That's a very less concentration. So at that concentration, it's effective. Imagine at higher concentration how effective it will be. Another is merthiolate. At a concentration, it is effective at 1:10,000 of dilution. So you can use it in very small quantity, and you can use it for preservation of the serum. So whenever you have to do check on patient's serum, whether if a patient is having allergy, you can keep the serum and then check for its IgE, because immunoglobulin E is responsible for its allergy. So you can keep the serum and see whether he has higher range of immunoglobulin. So if you want to preserve it, you can use merthiolate. Another is copper salts. They are used as fungicide. It means copper salts are particularly used for killing fungal infections on the skin. So disadvantages of this mercury chloride: it is highly toxic to our normals. To us, it means human beings, and they can be readily inactivated by the organic matter. Okay. Now, there are now surface active agents. So surface active agents are nothing but they are your soaps and detergents. Okay. So why do doctors force on using a soap and cleaning it for around 5 minutes or 5 to 7 minutes? Or in this corona time, they are also telling us to clean our clothes with good detergents, because detergents can kill any kind of viruses or bacteria. Because what detergents do is it leads to the lysis of the viruses. because our viruses have a surface membrane that's made of phospholipids. When you use detergents against them, so they immediately, disrupt and break the phospholipid membrane of many of the viruses or gram-negative bacteria. So what is their mode of action? As I told you, they can interfere with the lipid containing membrane, particularly present in your coronavirus also, and particularly in your gram-negative bacteria. For example, E. coli, or another example of gram-negative bacteria, as I told you, is a comma-shaped bacteria called Vibrio cholera, which causes cholera. Cholera means a patient has constant loss of, stool and water. Another is, as I told you, another example of gram-negative bacteria which can interfere with detergent is your Helicobacter pylori, Helicobacter. But particularly we will not use it because our stomach and the detergent will be toxic to our stomach. Okay. Surface active agents who have long hydrocarbon chains. Hydrocarbon chains are non-polar in nature. Okay. So they can intervene. They are non-polar in nature. Okay. So they are fat soluble and charged ions, and since they contain these features, so they can clean your surface membranes of these bacteria or viruses. They can disrupt membrane, as I told you, and it leads to the leakage of the cell contents. And this is how we achieve the destruction of microorganisms, by using detergents and soaps. So it results in your, it means it's like a balloon, and this balloon, the air comes out and that's finished. Okay. Examples: soaps and detergents. So detergents can be anionic, that's negative in charge, or they can be cationic, that's positive in charge. Cations are positive in charge and anions are negatively charged. So detergents containing negative charge with long hydrocarbon chains, which are hydrophobic in nature, they're called anionic detergents. Okay. So what it does is, if you have a stain on your cloth and you use detergent, it acts like a nail. Okay. When it acts like a nail, it removes the stain from your clothes, and this, because this stain is fat soluble, so it's because it's hydrophobic in nature, so these detergents cover it like this and removes the stain out of your clothes. So it's been covered by these detergents which have so this structure is called micelle, M-I-C-C-L-E. If you have studied plasma membrane structure, the typical example is RBC, because it doesn't contain any organelles, so RBC is used as a starting material for studying your membranes, that is phospholipid membranes. So this is how detergents work and remove the stains from your clothes, and it also kills the microorganisms in the same way. Okay. So detergents containing this negatively charged anionic detergents, and these include the soaps and the bile salts. In fat soluble part, if the fat soluble part is made up of positively charged, it can be done by combining with a quaternary nitrogen. As I told you, nitrogen has an ability to form coordination bonds. There are different bonds. There are covalent bonds, there are non-covalent bonds, and then there are coordination bonds. So non-coordination bonds are particularly, for example, hydrophobic interactions. They can be weak ionic interactions. They can be your hydrogen bonds, and they can be your weak. These are some examples of non-covalent bonds. So we call that detergent as cationic detergent if you treat it with quaternary nitrogen. Okay. So now we also have different kinds of dyes which can act as they can act as sterilizers, or they can disinfect different ways. So what is the mode of action? How do they work actually? So this some examples are like acridine dyes. There's a dye called acridine orange. Okay, it inhibits DNA. Acridine orange can be used to dye DNA. So, or prevents its replication. So it's bactericidal in function. It means it has a killing effect. Okay. Now, because of their interaction with the nucleic acids, that's either DNA or RNA, these can have an effect on the microorganisms. So one dye is aniline dyes, which contains amino group, that's called aniline dyes. They can be crystal violet, they can be malachite green, they can be brilliant green. So remember that you use in gram-positive bacteria, you use this dye for staining. Okay. And malachite green, it's a component of many growth medias. Same is the case with brilliant green. Now, acridine dyes such as acriflavin and aminacrin are two such examples of aniline dyes. Acriflavin is a mixture of proflavin and euflavin. So only euflavin has an effect, has an antimicrobial properties. A related dye called ethnium bromide is an intercalating agent. So if you have a DNA like this, okay, and it has bases, so it intercalates between the bases. Okay, so it prevents the DNA replication. So this ethnium bromide intercalates, and it can cause DNA damage. So it is a germicidal in action. Again, cidal is coming. Germ can be anything. It can be a virus, it can be bacteria, it can be fungi, but you should concentrate on the term cidal. It intercalates between the base pairs, okay, of the DNA, as I told you. So they are more effective against the gram-positive bacteria. Here, gram-positive bacteria means which have a thick peptidoglycan layer. Thick peptidoglycan layer. Examples I already told you before. They are Bacillus anthracis, your Bacillus subtilis, Bacillus. Then you have this Staphylococcus aureus, which causes MRSA. Okay, so these are a few examples of gram-positive bacteria. The only difference is they have a thick peptidoglycan layer as compared to the gram-negative bacteria. That's why when you stain them, they retain the dye. So they retain the dye is purple. Okay, as compared to the gram-negative bacteria. So in case of they are more effective against gram-positive bacteria, and they can act as a bacteriostatic. It means it only stops the multiplication of bacteria but doesn't kill it. But in case of gram-positive bacteria, it is very effective. So applications are, they can be used topically, that's on skin and mucous membrane. So they are antiseptic in nature to treat mild burns. They are used as a paint on the skin. For example, gum paints are there present for the inflammation of the skin, inflammation of the gums, and to treat bacterial skin infections or your mucous membranes, particularly your gums. The dyes are used as selective agents in certain selective media. For example, selective media means if you have a mixture of, if you have got some samples from forest, for example, soil, it contains different kinds of microbes. Okay, so let us consider there are different kinds of microbes, and you want only this microbe to come, so you need a selective media for it, in the media which can only grow this kind of a strain. Okay, so these dyes are used in these kinds of selective media. So now comes the hydrogen peroxide. So many of the patients who are again suffering from gum inflammation, they use hydrogen peroxide. So this peroxide can immediately relieve your gum pain. Okay, so this is one immediate way of getting rid of your gum pains. So it acts on the microorganisms through its releasing of the nascent oxygen, that is free radicals, that it releases superoxide anion. So these can have a very strong damage on your proteins and your DNA. So these are the free radicals. Example of free radical is superoxide anion. It can also be different kinds of anions, or for example N2 negative. So what are the applications of hydrogen peroxide? If you use it, and again percentage it comes, if you use it in 6% concentration, it acts as it decontaminates your instruments, it decontaminates your equipment. For example, particularly patients who are in the severe phase of COVID-19, they have to be put on ventilators. And before we put them on the ventilators, in order to prevent them from getting infection from other microorganisms, we can use this 6% of concentration of hydrogen peroxide to decontaminate the ventilators before we put the patient on the ventilator. So these are the precautions you people have to take, because you are the people who have to prepare the theaters. And if you use it in 3% concentration, it can be used for disinfecting skin, and it can deodorize the wounds, the ulcers. It has a strong sporicidal activity. Remember that. Hydrogen peroxide kills the spore-forming bacteria. The spore-forming bacteria are Bacillus subtilis. Few examples: Bacillus anthracis. So remember that they are active against the spores, which most of the are not. Other chemicals are not. So disadvantages is they decompose in light, so you have to keep them in brown bottles. So it can be also broken down by catalase, that is hydrogen peroxidase, which breaks the hydrogen peroxide. Catalase alternative is called hydrogen peroxidase. So it because of its proteinous nature, it drastically reduces activity. Its activity. So friends, so if we just conclude that this is the conclusion of my lecture of sterilization and disinfection by chemical methods. So remember, we have been studying it from the beginning, and last I will be talking about ethylene oxide. So please bear with me patiently. Okay. So now talking about the mode of action of the ethylene oxide, because it's a gaseous in nature, as we just studied it in the previous slides. Again, it's an alkylating agent. Alkylation again means you have to add this group CH3 C double bond O that provides a negative charge on the surface of your microbial proteins or their surfaces. So it alkylates this sulfur-hydryl group. It can alkyl the amino group. This is this. Totally they are present in the amino acids. Carboxylic group, that is COOH, and hydroxyl groups, that is OH. What are the properties of ethylene oxide? It's gaseous in nature, it's colorless, and it can also appear in the form of colorless liquid. It's cyclic in nature and colorless at room temperature. It means if you increase the temperature, it will change into the gaseous nature. It is a sweet ethanol odor, and you can identify ethanol oxide by this. And it can readily polymerize and is flammable. So always be careful while using ethylene oxide. Applications are: it is an effective chemisterilant. Sterilant means no growth of any bacteria, completely sterilized, killing everything, spores and gram-negative bacteria, gram-positive bacteria, funguses, viruses. So that's what I told. So since it's highly inflammable, you have to be very careful. But in this case, if you want to be on the safer side, you can use it in a combination with carbon dioxide. Different concentrations can be used, and it can, or you can convert it into dichloromethane, which is not flammable. It requires presence of humidity. Without humidity, it cannot work. So you have to provide the free air to it, and it has a good penetration. It can be well absorbed by the porous materials. So it's used to sterilize heat liable materials. Heat liable materials are the materials which you cannot put on the you cannot sterilize them on a high heat. Okay, so you can use it on the beddings, the textiles, the rubber part, the plastics, the syringes you use in the hospitals, disposable petri dishes which are made up of plastic again, complex apparatuses like heart-lung machines, respiratory and dental equipments. So you can sterilize them by using ethylene oxide. So efficiency test is done by Bacillus subtilis var niger. So it's a bacteria, a spore-forming bacteria. So if it kills this, so it means your sterilization is complete. So you can use it as a positive control. Positive control. So what is positive control? Positive control is the one which that will ensure you that your sterilization is okay. Now there are disadvantages, as we have discussed with every disinfectant or antiseptics. It is highly toxic, irritating to the eyes, skin, again highly flammable. You have to be careful about it. And another problem is mutagenic and carcinogenic. It can cause cancer. So you should be very careful about the exposure of ethylene oxide. So another one is the beta propiolactone. You might have heard about the bacteria propionobacterium. It causes acne in humans. So it has the same name like beta. It has a beta structure, propionolactone. So it's a lactone in structure. It means ring structure. Again, it acts as an alkylating agent, and it does it on the carboxyl groups, that is C double bond OH of the amino acids, hydroxyl groups OH. Properties: it is a colorless liquid with a pungent smell, slightly sweetish. It is a condensation product of ketane plus formaldehyde. So it is a combination product. Applications is it's an effective sporicidal agent. Remember that in your chemical sterilizers you have to remember which of the chemicals are effective against spores. Okay, and it has a broad spectrum activity. It means it can act on many different microorganisms, whether it's gram positive, whether it's gram negative. Again, using concentration, if you are using it at 0.2, it means around half an 1/4 of a concentration. So 0.2% it can be used to sterilize biological products. It's more effective in fumigation, as we told about. We used for fumigation we used to have formalin, and then we can mix it in potassium permanganate or heat it, steam it, and you can sterilize your culture rooms and biosafety hoods. So it's used to sterilize the vaccines. There's no vaccine right now currently available for your coronavirus. That's why we are suffering like this. It can be used to sterilize the tissue grafts. For example, tissue grafts are particularly used for in the skin burns. And you can also use it to sterilize the surgical instruments. Now, again, we have advantages and disadvantages. So it has the disadvantage of this beta propiolactone is it has a poor penetrating power, and again it is carcinogenic. It means it can cause cancer in humans. So based on if I try to conclude this lecture, so based on the need of the sterilization or disinfection, we can use different kinds of chemicals. We can use both physical methods and the chemical methods, and lastly we will talk about the physiochemical methods. So what is a physiochemical method? Simply you can use formaldehyde and you simply heat it so that the steam is formed. So for example, how is it done? Let us see. So its mode of action is physio as well as chemio, and chemical plus physical. So adopts both the methods. So what you do is you steam the formaldehyde. This is the physiochemical method of sterilization. It means killing the bacteria, acidal, which takes into account the action of the steam, which we studied in the physical methods of sterilization, and here as well as using the formaldehyde. So saturated steam is being first produced at a pressure of around 263 mm per square inch. You increase the temperature to 70 degrees Celsius. The air that is present in the chamber is removed from the by using is flushed, and that's flushed in is removed, and then saturated steam is being flushed in. So steam is there. Now there's then step number 2 is adding formaldehyde. So you can inject formaldehyde into the steam in a series of pulses. Pulses means you have to inject and stop, inject and stop. So each pulse will last for 5 to 10 minutes, and the articles are put at a holding temperature. It means temperature has to be constant around 70 degrees Celsius. Okay, and the time is for the sterilization is 1 hour. In autoclave, the time of sterilization, you should remember the pressure is 15 per square inch. For 15 minutes you have to keep in autoclave, but this is a different thing. Okay, so formaldehyde is then flushed by out inflow of the steam. So what is the disadvantage of using this steam physiochemical method is it causes the condensation of formaldehyde can take place, and this can produce a large induction of formaldehyde that wets the steam, resulting in loss of latent heat. So when you talk of heat, this is a physical method. So if you lose this heat, because of this condensation, so it will not work. Another thing is, again, you can use a positive control to check whether your sterilization is complete or not. You can use a paper strip that has the logarithmic number of spores on it, and this is the spore name is Geostereothermophilus. It means this kind of bacteria can they are very temperature, they have high ability to retain the or to grow in the high heat. Okay, so this is the end of the lecture, so I will stop it here. So this is the end of the sterilization and disinfection, where you have studied all the methods, physical methods and the chemical methods and the physiochemical methods. I divided it into two parts. Part 1, part first talks about physical method, part 2 also talks about physical method of sterilization, and part 3 talks about chemical and physiochemical method of sterilization. So thank you for patiently bearing with me. What I require from you is your feedback. Feedback includes your comments, it can include your questions, it can include your wherever you feel a need to contact. You can contact me on your WhatsApp group, or once I upload it on YouTube, you can put your comments over there. So thank you very much, and goodbye. Take good care of yourself.